Hello, everybody, and welcome back to BioSC 140 Human Physiology. This video will be the experimental procedures video for Lab 23, which covers white blood cells. Pop quiz. What is another name for a white blood cell? If you've been following along in lecture, you should be ready for this question. Another name for a white blood cell? Leukocytes, or WBCs, is another way you'll see it. All right, so there's a few procedures that we're doing in this lab. Some parts are similar to the red blood cell lab, and some parts are different than the red blood cell lab. So we're going to be doing a total white blood cell count. The procedure is similar to doing a total red cell count with a few changes. I'll make sure I point out those changes. Then we're going to do something called a differential white blood cell count. All right, so Fox 6.2 or Lab 23. All right, so a little background information. Blood is composed of liquids and solids. Blood is about 60% liquid. The liquid part is called plasma. Now, we have talked about hematocrit. We have talked about how that value can change from time, or does change from time to time, and has a normal range. The liquid part is made up of water and the dissolved stuff, which is ions, nutrients, gases. The solid part, around 40%, 38, 40%. The solid part is made up of three main things: erythrocytes, or red blood cells, RBCs, leukocytes, white blood cells, WBCs, and also the answer to the pop question, the surprise question I just asked, and thrombocytes, or platelets. They are clotting cells. Most formed elements are red blood cells. About 95% are red blood cells. There's around 5 million red blood cells per microliter. Leukocytes, or white blood cells, there's around 5 to 10 million per microliter. And thrombocytes, or platelets, there's around 200,000 to 400,000 per microliter. So right here, this image, we have it broken down a little bit further. So we have whole blood, which is composed of plasma, the liquid part. The liquid part is made up of mostly water, and then the dissolved stuff. So ions, organic molecules, trace elements and vitamins, gases. Gases, we usually talk about CO2 and O2 when we talk about the dissolved gases. The organic molecules are going to be your amino acids, your proteins, your glucoses, your lipids, nitrogenous waste. For the cellular elements, or the formed elements, we're going to have our erythrocytes, or red blood cells. We're going to have our platelets and our white blood cells. Now, there's many, many types of white blood cells. There's 5 that we focus on in this class, 5 main types. We've got our lymphocytes, our monocytes, our neutrophils, our eosinophils, and our basophils. All right. So last time, Lab 22, we mainly talked about these red blood cells, RBCs, erythrocytes. In this lab, Lab 23, we're focusing on the white blood cells, the WBCs, today's lab. Safety first. So we are in a virtual setting right now. We are not performing this experiment live, but I still think it's important to go over precautions, safety procedures, because one day we might be back in lab. We'll see. All right, so this is a blood lab. Whenever you're handling somebody else's blood, use universal precautions. That means you assume all material is hazardous. You assume it's hazardous, so you treat it as such. You wear gloves, especially when working with the blood of others. Properly dispose of and clean contaminated materials. Lancets are what we use to, we've talked about them in past labs. That's what we use to put a little tiny hole in the side of our finger to get a sample of blood. So they are sharp, and any other contaminated sharp or hard objects we put in the sharps biohazardous material container. Soft materials, soft contaminated things like alcohol wipes, gauze, band-aids, paper towels go in the soft biohazard bag. Reusable contaminated equipment gets washed thoroughly with AMFO. Do not wash blood contaminated equipment for anybody else. If your blood is on the equipment, you should wash it. No eating or drinking in the lab. If you're hungry, please step into the hallway. All right, so Fox 6.2. This is going to look really similar to how we did the total red cell count, but this is the total white cell count. So first, we must obtain a small capillary tube full of blood. It looks like this. Looks like this. The ones we use in lab are about an inch or so long. You need to fill it up completely with no air. We are going to be doing some math, some dilution factors, and to get an accurate answer, you need to fill this up with no air bubbles completely. We are then going to take that capillary tube and we're going to put it in a tube that looks just like this. And this tube is going to contain a solution that is going to lyse every cell but white blood cells. So red blood cells get lysed, you don't see them anymore. We're going to shake it, let it sit for a minute, and get all of the liquid out of the capillary tube. We are then going to go and use the hemocytometer. Now you should know this word from our last lab, hemo blood cytocell meter counter, so a blood cell counter. Let's go to the next slide so we can see our hemocytometer. We're going to add drops to these little V-shaped indentations on the side, and then we're going to add a cover slip. There are special cover slips for the hemocytometer. Everything up to this point has been pretty much the same as what we did for the red cell total count, but this is where it's going to change. The squares that we count are different. The squares that we count are different for the white cell lab. We're going to count this big square, this big square, this big square, and this big square. Expect to see 25 to 50 cells per square. If the cell is half on the top line or the left line, it counts as inside the square. If the cell is on the bottom line or the right line, it does not count as inside the square. Add up A, B, C, D, and times it by 50, and you should get your total white cell count. This is what it'll look like. Perfect. So the two main differences between the white cell and the red cell are where you count on the hemocytometer, the squares you count, and then the conversion factor. It's 50 for the white cells. All right, next up, we're going to do our differential count. So the goal of this is to get a differential white cell count. What a differential white cell count is, you might actually remember back to the first day of class when you did the math for a differential white cell count. It's you take all the white blood cells as a whole, what percentage are neutrophils? You take all the white blood cells as a whole, what percentage are basophils? It's what percentage of the whole is each different type of white blood cell. So what we're going to do is we're going to prepare two slides, one as a backup. We're going to place a drop of blood on the end of a clean slide, about 80% towards one side, like this demonstration. We're then going to take a clean slide. We're going to take a clean slide and we're going to slide it to close the end and touch the drop. So basically we're going to have that drop of blood on our slide right like this. We're going to take another slide, a clean one, and we're going to hold it at an angle. And we're going to slide it along the slide until we touch the sample of blood. And when you touch that sample of blood, it's going to spread out across horizontally, like right here. It's going to spread out horizontally across the end of that cover slip. We are then going to push that cover slip across the long way across the other slide. Sorry, I just said cover slip, but it's actually another slide. We're going to push it the long direction across the other slide. And what we're going to get is we're going to get a smear of blood across the slide. This is really satisfying, and I wish we could do it. Students always have a ton of fun doing the blood smear on the slide. So drop of blood near one side, 80% towards one side. Cover slip at, sorry, not cover slip, slide at an angle, drag it across, like right here. You can see right here the angle. Drag it right here. You can see the angle. Drag it across the slide until you touch the blood. Once you touch the blood, the blood spreads out across the slide. You then smear it back long ways across the slide. And you allow it to dry. You then take it over to the stains. We're going to dip it 5 times in solution 1, which is a fixative, 5 times in solution 2, which is a stain, and then 5 times again in solution 1. We're going to rinse and let it dry. We are then going to identify, we're going to count 100 cells. We're going to count 100 cells. So I don't expect you to be perfect, and we're not going to be perfect at identifying the different white blood cells. This is our first time doing it. We're just being introduced to it. But, you know, looking at the photos provided, knowing the morphological characteristics of each white blood cell, you know, students do their best to visualize and count the different types of white cells. Now, we count 100 cells, 100 white cells, because it's really easy to do math. If you count 50 neutrophils and you count out of 100 total cells, it's 50% are neutrophils. If you count 64 neutrophils and you count 100 cells, 64%. So that's why we do 100, and it gives us a pretty accurate differential. One more thing I want to point out, these counters up here. So we actually use these in lab, and each one of these buttons is labeled. You push this button when you see a neutrophil, this button when you see an eosinophil, this button when you see a monocyte, and so on. And so it'll count each, it'll count each type that you see, and it'll also have a total counter also. So you can see the total number. Really helpful. Safety again, everybody handles your own blood, dispose of everything properly, and I'll see you in the next video for discussion.